Hi all, welcome to Physio Exam Buddy. In today's video, we are going to learn the carbon dioxide transport in the blood, a very important and frequently asked topic in the MBBS physiology. We will focus on the three forms of carbon dioxide transport, their mechanisms, and the important concepts you need to remember for your exam. Carbon dioxide, it is formed inside the cells mainly by the aerobic metabolism of glucose. As a result, the intracellular PCO2, it will be 46 millimeters of mercury, and that of the interstitial fluid surrounding the cells, it is 45 millimeters of mercury. So, the carbon dioxide will diffuse from the cell into the interstitial fluid against a concentration gradient of only 1 millimeters of mercury, that is 46 minus 45, that is 1 millimeters of mercury. And from the interstitial fluid, the carbon dioxide diffuses into the blood flowing in the tissue capillaries having a PCO2 of 40 millimeters of mercury against a gradient of 5 millimeters of mercury. Average 4 ml carbon dioxide will be transported from the tissues to lungs per 100 ml of the blood. Carbon dioxide is transported in three forms. The first one is dissolved form. Around 7% is transported in the dissolved form, 23% as carbamino compounds, and 70% as bicarbonates. First of all, we will see the dissolved form. Carbon dioxide, it is 20 times more soluble than oxygen. So 0.06 ml of carbon dioxide will be dissolved in 100 ml of the blood for each millimeters of mercury pressure. So the venous blood with PCO2 of 45 millimeters of mercury, it contains around 2.7 ml per deciliter of the carbon dioxide in the dissolved state. Whereas the arterial blood, the partial pressure of carbon dioxide is about 40 millimeters of mercury, and it contains about 2.4 ml per 100 ml of the carbon dioxide in the dissolved state. Thus, only around 0.3 ml, the difference is 0.3 ml of carbon dioxide will be transported in the dissolved state per 100 ml of the blood from tissues to the lung. This represents around 7% of all the carbon dioxide transport. Please remember all these values because these values are taken from the international textbooks. The second form of transport is by carbamino compounds. That is, after entering the blood, some of the carbon dioxide, it will combine with the amino group of the proteins to form carbamino compounds. And in the RBCs, this carbon dioxide combines with the amino group of the hemoglobin to form carbamino hemoglobin. And the deoxyhemoglobin forms this carbamino hemoglobin more readily. And this combination of the carbon dioxide with the hemoglobin, it is a reversible type of reaction that occurs with a very loose bond. So the carbon dioxide is easily released into the alveoli where the PCO2 is lower than that in the pulmonary capillaries. In the plasma, the carbon dioxide combines with the amino group of the plasma proteins. But this reaction is very less significant and also very slow reaction. So in short, a total amount of 1.5 ml per deciliter of carbon dioxide will be transported in the form of carbamino compound. And the main form, that is the bicarbonate form, that is at the tissue level, most of the carbon dioxide enter the blood will diffuse into the RBCs. And inside the RBC, it is rapidly hydrated in the presence of the enzyme carbonic anhydrase to form H2CO3. But H2CO3 is a highly unstable compound, so it will soon dissociate into H plus and HCO3 minus. And when this HCO3 content of the red blood cells become more than that of the plasma, about 70% of the bicarbonate will leave the RBC and enter the plasma. H plus cannot move out because the RBC membrane is impermeable to the cations, and the H plus, it will be the H plus that is formed is buffered by the deoxyhemoglobin, and this buffering of H plus is very critical to keep the reaction moving towards the synthesis of HCO3. These bicarbonate ions then leave the RBC and enter the plasma in exchange of the chloride ion for maintaining the electrical neutrality. And this exchange, it occurs through a membrane protein in the RBC membrane, which is known as the anion exchanger 1, also known as AE1, formerly called as the band 3 protein. And this exchange of bicarbonate for the chloride, it is known as the chloride shift or the Hamburger phenomenon. And because of this chloride shift, the chloride content of the red cells in the venous blood, it will be significantly greater than that in the arterial blood. And this reaction is very, very rapid and completed within 1 second. When chloride shift is asked, you have to draw this diagram also. And for each carbon dioxide molecule which is added to the red cell, there is an increase in the one osmotically active particle in the cell, either a bicarbonate or chloride in the red cell. Consequently, what happens is that the red cells can take up water and increase in size. Because of this, the RBCs in the venous blood has highest osmotic fragility, which can be again asked as a physiological basis question. And in the lungs, what happens? The reverse happens. That is, the carbon dioxide will be given out and chloride will move out of the cell by a process which is known as the reverse chloride shift. So this is the diagram you have to draw when the carbon dioxide transport is asked. That is 7% of the carbon dioxide it is transported in the dissolved form, and as carbamino compound 23%, and as bicarbonate the rest 70%. And applied physiology for the transport of carbon dioxide, you can write it as a drug named as acetazolamide. It is a carbonic anhydrase inhibitor. So whenever this drug is given, the carbon dioxide transport will be poor and the tissue PCO2 can even rise up to 80 millimeters of mercury. Next thing you have to write along with the carbon dioxide transport, it is the Haldane effect, which can also be asked as a short question. That is, the binding of oxygen to the hemoglobin, it will reduce its affinity for the carbon dioxide, and this will tends to displace the carbon dioxide from the blood, and it is important in promoting the carbon dioxide transport. And the Haldane effect results from the fact that the combination of oxygen with the hemoglobin in the lungs causes hemoglobin to become a stronger acid, and the reaction is this thing. And this will displace the carbon dioxide from the blood and into the alveoli in two ways. The first way is that whenever there occurs an increase in the acidity of the oxyhemoglobin, it causes it to release an excess of H plus ions, and these will bind with the bicarbonate ions to form the carbonic acid, which soon dissociates into water as well as carbon dioxide. And this carbon dioxide, it will be released from the blood into the alveoli and finally into the air, and thus oxygenation of the hemoglobin in the lungs increases the carbon dioxide unloading. And the second way of the Haldane effect is that the acidic oxyhemoglobin, they have a less tendency to combine with the carbon dioxide to form carbamino hemoglobin, thus displacing much of the carbon dioxide that is present in the carbamino form from the blood. And the deoxyhemoglobin, which is present in the tissue capillaries, is capable of loading more and more amount of carbon dioxide than oxyhemoglobin because of again two reasons. That is, deoxyhemoglobin binds with H plus actively than oxyhemoglobin, and the second one is that the deoxyhemoglobin have more affinity for the carbon dioxide. Now we will see what is this carbon dioxide dissociation curve. This curve just gives the relationship between the PCO2 as well as the total carbon dioxide content in both the arterial as well as the venous blood. So here the blood is exposed to various carbon dioxide tensions and the corresponding carbon dioxide content will be determined, and the graph is plotted with PCO2 on the x-axis and carbon dioxide content on the y-axis for both the venous blood as well as the arterial blood. And this curve you can see here, it is more linear as well as more steeper when compared to our oxygen dissociation curve, and at any given PCO2, the reduced blood, that is the deoxygenated blood, they contain more carbon dioxide than the oxygenated blood, and the carbon dioxide dissociation curve is affected significantly by the partial pressure of oxygen. This is again the carbon dioxide dissociation curve, and 2 points were marked here, that is point A as well as point B. Point A in this graph, it denotes the venous blood in the tissues, and the PCO2, it is around 45 millimeters of mercury, and the corresponding carbon dioxide concentration, it is 52 ml per deciliter. Whereas here the point B, it denotes the arterial blood, where the PCO2 it is around 40 millimeters of mercury and the corresponding carbon dioxide concentration it is around 48 ml per deciliter of the blood. So what is the difference here? It is 4 millimeters, the 4 ml, that is 4 ml of carbon dioxide will be transported per 100 ml of the blood. So you know that the cardiac output it is 5 liters. So the carbon dioxide transported can be calculated by 4 into 5000 by 100, that is 200 ml per minute. Now we will see what is the significance of the Haldane effect, which helps in the transport of carbon dioxide from the tissues to the lungs. So due to Haldane effect, higher the saturation of hemoglobin with oxygen, lower the carbon dioxide content for the given PCO2. And if there is no Haldane effect, the carbon dioxide content would fall only to 50 volume percentage. Loss of only 2 volume percentage of carbon dioxide will happen. In the lungs, when the partial pressure of oxygen increases, it lowers the carbon dioxide curve from the top to the bottom curve. That is what we have seen in the previous graph, point B. So the carbon dioxide content will fall to only 48 volume percentage. The additional 2 volume percentage loss of carbon dioxide. So thus the Haldane effect, it actually approximately doubles the amount of carbon dioxide released in the lungs and approximately doubles the amount of the carbon dioxide pickup in the lungs. So that is about the carbon dioxide transport. Thank you.